regarding the Cot Cup, we will discuss. My name is Dr. Sujit Ghosh from JK College, Purulia. Uh, in the Cot Cup, we will discuss today what does Cot means, meaning of denaturation, meaning of renaturation, factor responsible for renaturation, steps in Cot analysis, analysis of Cot curves, and significance of Cot curves. What does Cot mean? The Cot means analysis of rate of renaturation of DNA sample. In the figure, you can able to see that it is a double-stranded DNA. Upon giving heat and alkali, this double-stranded helical stacked DNA becomes single-stranded, and this single-stranded strand is called as denaturated state. From denaturated state to renaturation can be again be done by using the by lowering down the temperature and neutralizing the alkali. And what does denaturation means? The denaturation means process of formation of single-stranded DNA through heating or through application of alkali into single-stranded DNA. Here, main purpose is to breaking down the hydrogen bond which is present in between A double bond T and G triple bond C. Here we find that there is an increase in absorptions of U whenever we applied UV 260 nanometer wavelength of light, and this is called hyperchromic effect. The rate of increase in absorbance is directly proportional to rate of denaturation. Upon denaturation, viscosity of the DNA decreases. And what is renaturation? The renaturation is the process of formation of double-stranded DNA from a single-stranded, denatured, complementary DNA strand. Means it is again the formation of stacked double-stranded DNA. It is effected by cooling or by withdrawing or neutralizing the alkali. It involves formation of hydrogen bond. Here, decrease of absorbance upon renaturation. Upon renaturation, viscosity also increases. This can be detected by using UV spectrophotometer absorbance at UV 260. Now, what are the factors responsible for uh, renaturation? DNA concentration, time, the temperature, and the cation concentration in the wafer, as well as viscosity. What are the steps in Cot analysis? Firstly, we have to isolate the DNA from the cell, and this DNA is cutted at 1 kb in length. The fragment is completely denatured by using the temperature. The melting temperature have to be calculated, where 50% will be DNA will be melted. The fragments are then again allowed to renature just below the 10 degrees centigrade of melting temperature. Now, renaturation can be studied whether it has taken place or not by using spectrophotometer at an absorbance of 260 nanometer. Single-stranded DNA absorbs more light efficiently than double-stranded. And another way is to using hydroxyapatite chromatography. In this hydroxyapatite chromatography, crystalline calcium phosphate are being used, where double-stranded DNA can be absorbed easily but single-stranded not. Then comes what is then C Cot. The Cot is the concentration of DNA in mole per liter. renaturation time in second, that is indicated by T, and the buffer factor, uh, which is giving a giving a indication what amount of cation is present. Now, whenever we analysis it is a graph. In x-axis we plotted log of cot, and on y-axis percentage of single stranded DNA. Here we get a result of three area. The first one immediately being immediately being renatured, and this immediately being renatured is called rapidly renaturing. It is not as rapid as that, so it is immediately repetitive, and it is also very late, so it is single copy. That means the rapidly repetitive DNA sequence showing concentration than the other within the genome, and a part of the genome consists of highly repetitive sequence. It is the concentration is moderately repetitive, and the concentration here is very less repetitive. The last one, but why do we uh, analyze Cot curve? The Cot curve is the analysis for particularly to know the kinetic complexity of genome, estimation of genome size, estimation of the proportion of single copy, moderately repetitive copy, and highly repetitive copy. It also gives us a relationship whether whenever we compare two Cot curves, for example, from one bacteria from another bacteria, it will give us whether they are similar or dissimilar in nature. Thank you.